Hello everyone, this is Alan Wu. I'm from Bio 230. This is part of my Ecology in Your Life project. And this is Delta Nature Reserve, which is also Fern Spot, which is part of it. It's just down the trail that way, going by the Northern Overpass. And on a trail, which last time I came here for, you see a lot of local vegetation species, including hardhack, including salmonberry, salal, all sorts of vegetations that are really interesting. But amongst those, there is the Himalayan Blackberry, which is also called Rubus armeniacus. And it is an invasive species non-native to here, and it basically takes a lot of space up. Through competition, it outcompetes local species such as Western Hemlock, a lot of the shade intolerant species by taking up the space, and also outcompeting the deep-rooted plants. And this is usually really detrimental to the local area because it makes eroded lands area because deep-rooted plants cannot thrive in the local area anymore, as well as it makes the place basically relatively flammable, vulnerable to fires and such. So my main ecological question in my Ecology in Your Life lab will be, what is the biotic interactions between Rubus armeniacus, Himalayan blackberry, and other local species in the area? To do that, I will use my ruler and a tape measure. I will find a Rubus armeniacus specimen, and I will lay out 10 meters to either side of it down a trail. I will not go inside the trail, but rather just along the trail. And 10 meters to both sides, and I will use a 1 meter by 1 meter quadrant, and inside each, I will count the relative abundance of each focal species in the area. I am hoping to find that as it gets further away from the Rubus armeniacus specimen, there will be a greater abundance of other specimens. So right now here is an actual Burns Bog Delta Trail boardwalk, right? So we're actually currently inside the very inner part of the trail, essentially. And here, you can see along the sides that there are basically no occurrence of Himalayan blackberry or Rubus armeniacus. Reason being is probably a very likely chance that they want to remove it directly and keep it as low as possible to conserve the local area of the focal species. You can see example of focal species such as this. This is salal. Salal is it feels like this texture, right? You can tell by its waxy leaves, and its berries, although it's a little bit dried up now, are black. And natives really value salal because they harvest them, and it's important to their diet, and it's part of the culture as well. So salal can tell by the waxy leaves, and is part of the focal species here. And another species, if you just come further down here, or that way actually. Here is Western Hemlock. It's a relatively young tree. You can tell it's a Western Hemlock by its blunt edge leaves and of varying length, and they're quite flat. So these are Western Hemlock here. Also known as Pseudosuga menziesii is Western Hemlock. Now, if you come over to this side, down here is also another species here, focal species. It is deer fern, right? You can tell it's a deer fern because it doesn't have serrated edges like the sword fern, and the leaves are all quite flat, and they're alternatively on the stem, like the central stem right here. So here is a deer fern right here. It's a very young one. Yeah, so these are all very interesting focal species and local here at the Burns Bog Delta Reserve, and we see a lot of them here, but we don't see the Himalayan blackberry in this area. This railway divides or sets a boundary for the Delta Natural Reserve Burns Bog area, right? So on the other side here, it's all Burns Bog Delta Natural Reserve, But once past this way, going further up, there's going to be a linear trail, which is going to be North Delta Greenway. So we will be heading up there, collect our sample, and talk more about the relative abundance of species relative to a baseline sample species of Rubus armeniacus or Himalayan blackberry. Alright, now we are on the North Delta Greenway trail. So we're already kind of further away from the Bogs Burn Delta Nature Reserve. So here, right now, behind me is an entire thicket of Rubus armeniacus, right? You see the specimen right here? It is Himalayan Blackberry Rubus armeniacus. You can tell by its prickly spine, right? Right now it's a young one, but if you come over this side a little bit more, you can see older specimens with its red, like more red and brown stem right here, right? It's prickly, right? It has 5 leaves, 1, 2, 3, 4, 5, and 5 lobes in this first year, and the second year, which it grows the blackberries that we normally see, it will have 3 leaves. It's not local to the area actually. People suspect it came from Eurasia in 1870s, and it's very invasive and spread really fast. So it outcompetes deep-rooted plants such as ponderosa, red alder, and such, right? And by doing that, it creates land's erosion sites, right? Making the soil very prone to erosion by water and flooding and all that such. So it's very bad for the local habitat. As you can see, the Bogs Burn Nature Delta Reserve, we don't see much of them, but here you see the entire thicket from all the way here, all traced to around 5 to 6 meters that way. So for that reason, I doubt this will be a good specimen as a baseline to go either way, because it's just taking up such a big area in one way. But it helps us characterize the nature of Himalayan blackberry in that it's just very, very abundant, and it just outcompetes a lot of the other species and growing really rapidly and spreading like wildfire. On that side is where the Delta Nature Reserve Fern Bog area is. So you can see that side of the railway, basically not a whole lot of Himalayan blackberry. But on this side, where it's not protected by Natural Delta Reserve, you see them along the path quite abundantly. So we're going to move forward and keep looking for our baseline specimen of Himalayan blackberry to do our data on. Great. So if you will come over here, here is a good specimen I'm going to use off of as baseline. So right over here, you can see this is a Himalayan blackberry sample by its prickly stem Eiyl. I'll use this 1 meter. Here I set it to 1 meter ruler. Right here, facing off of this berry, one of this decayed berry right over here, using this as a baseline for 1 meter. So from here to the left side 1 meter will not count in my sample. It's going to be just the baseline. From here it counts as 0. 1 meter to the right side of this will count as 1 meter, right, to the right of it. Now back to the baseline. To the left of this 1 meter baseline is going to be -1, which will count as the left side of the baseline. I will do this 10 more times or 9 more times on either side. So it will be -1, -1, -2 meters, -3 meters, -4 meters, and so on. For 10 meters in total that way to -10. Also on here, right, baseline to the right side of the blackberry, 1 meter, again 2 meter, 3 meter, all the way to 10 meters that way. For each meter quadrant, what I'll do is place my ruler inside like this, just over the top, right? Look over the quadrant, and look at the percentage cover, estimate it. So I'll do this for every quadrant, and yeah, let's get on with the data sampling.
So welcome back everyone. Right now I have taken back all the photos I've at the site. Here is my sample specimen. You can tell by the little dried up berries in the bottom right. From there, I use an Excel file. You can tell I put the data in already. At 0 point, it's not shown on the graph because it's assumed to be 100% abundance at 0, serving as a control. To the right, here's 1 meter. You can pause the video for each photo if you want to take a better look, comparing that to the data here. So here is 1, here is 2, 3, 4, 5, 6, 7, 8, 9, 10. Alright, this is 10. Here is a picture of me laying out a quadrant. And here's a picture from where I laid out from the sample specimen to the right of the sample quadrant at 0 point. And here's to the left, -1, -2, -3, -4, -5, -6, -7, -8, -9, and -10. Right, and here is a comparison. You see in the photos, 1 meter around from my elbow, which is 40 centimeters, and the ruler itself, the middle one, is 60 centimeters, so that adds up to a meter. Alright, yeah, so that is it for the photos. And now, for the data analysis, for the Excel file, you can tell that I've already made a graphs for these. I'll make the first one, which has like a complete Y and X axis titles. I did not do for the other ones because it's a little bit redundant. You can tell already they're all like the same, similar kind. I just repeated what I did for Himalayan Blackberry across all others. I tried to use a bar graph, but the scatter plot is the only thing that can really compare the data between the -10 to 10 meters and this, laying out nicely. Other bar graphs does this funky thing where it doesn't really show comparison. I'm going to quickly show you. If I do this, insert, and try using a bar graph to display, it does this weird thing where it doesn't really show the position. Yeah, relative area. You can take a look at the graphs, pause if you need to. So yeah, from these graphs, I made a few trend notices. To the right side, Himalayan Blackberry lowers faster, does a quick dip. To the left, it remains in medium abundance, going downwards. And the main finding is that it does have a little bit of biotic interaction to other vegetation species, such as trailing blackberry, where we see increasing in relative abundance of trailing blackberry as it gets further away from the center, to the right and also to the left. There's also a little bit of biotic interaction between Himalayan blackberry and creeping buttercup, sword fern, and spiny wood fern. As you can see by these graphs, a little bit further away to the left from a central specimen is going to increase a little bit relative abundance, increase a little bit of relative abundance. Yeah, one of the false assumptions is that I'm going to this by biotic interactions, but another possibility is that they have already dispersed there. The original specimen has already dispersed there, making it that there isn't space there for the other species to go there, which is also a abiotic requirement. Part of a space, right? Sunlight, soil, space, shade, water, they're all abiotic requirements these vegetation needs, and that also brings back to biotic interactions as well, because the original specimen, sample specimen, I said at 0 mark, Himalayan blackberry might have taken up the space, and where the other vegetation cannot get to there, already. Yeah. Yeah, the facilitation I'm talking about here is to the tree to the right from its shade for English holly and trailing blackberry. Here at around 4 to 5 meter mark, and you can tell from my Google Photos here, there's a tree at the 4 meter and 5 meter mark. I do not know the identification of that tree, but there seems to be a greater abundance of English holly around this area. So I suspect a little bit of facilitation there, but that is not really important to my ecological question. But that's a cool reference to go off of there that relates to the lecture material. So all in all, my ecological question of what is the biotic interaction between Himalayan blackberry and other local vegetation species, it has been answered partially by my Excel data from my photos and my analysis, and also completely by looking at these photos and looking at the graphs and comparing them to each other. I really hope you enjoyed my presentation, and yeah, thank you for listening and tuning in to me.
Hello?
Okay, and how did...
I got it!
Okay.
Oh yeah, so yeah.
Alright, disperse.
Let me do my
Okay,
task.
Let me just do my task first.
Disperse.
I'm going to reactor.
Bye, guys.
Ah, the plants are dispersing now. I'm coming to.
Can we hide? We can hide. Or we can hide behind the plant.
I am
I'm in
coming.
my own niche. Come and find me.
Oh. Oh, you got something to show me there?
Dang it. Okay, I guess I have to stay in the room when I'm a ghost too.
No, it's fine. Oh. Give me your nitrogen. I'm here to take your water. Oh.
Yeah, my water. No.
Oh.
Not my water.
Yum. Oh. Oh ho. Mind if I extend my roots over there?
Oh. Yo, we should do this for, what is it? Bio 260 next semester? Plant physiology?
Yes. Oh ho. Oh ho. Ah, trail blackberry.
No.
I know we're both blackberries.
You don't have to do this.
Only one blackberry can remain here. Goodbye.
Oh man, you're doing so well. Does that mean I get to keep my niche?
Oh wait, you survived.
I was hiding.